Today, we are going to explore a bit about the chromatin accessibility analysis for identify high confidence peaks. That is a way to say that we want to select peaks that are highly correlated with genes, so they are suitable for run different kind of analysis. And this is, let's say, a core part of the analysis or a core step when we have to define not only the strategy, but also the steps into the strategy to target our main goal in this kind of analysis. So first of all, I'm going to share with you this document. In this document, I try to gather most of the relevant topics that I have selected from different resources, because to be honest, I have found that this kind of step that is very relevant, it's poorly documented or is very specific, so we cannot just take these pieces of code and try to reproduce a specific pipeline, because it is driven for specific goals. So here at background, this is just one page of notes when I have tracked different kind of topics. The background for this is just for this talk is to focus on strategies and resources for this downstream analysis that is focused on single-cell ATAC-seq data. And as I said, the goal is just to gather resources and in a way that you have different kind of sources to explore or deep on the strategies to identify these kind of relations. Some key papers that I have dragged in the literature that, let's say, in the case of the human brain, better describe the steps. I think that could be these three. So you can explore later in more detail the steps specific for different kind of strategies, different goals. So I think that is very useful when you are trying to define a strategy. Also, I have here a couple of vignettes. These vignettes are more oversimplified versions to do something, but they don't provide indeed the fine details that we need to do this kind of analysis, but I could say that it's a good starting point. So if you want, for example, to check in data structures of how is the object interaction in the chromatin accessibility data, here I have add links for each one of these specific topics that are very relevant to understand, let's say, the conceptual approach, but also what are the main functions related with these different steps. I will not go into each one of these links. These links have been provided just for let you later to explore different kind of resources that you can have for clarify doubts or just follow one of these strategies. Also, I have added here core functions and references that I have using in most of my pipelines. So most of the analysis that I'm going to present is focused on Signac as the main object that is wrapping RNA profiles, but also accessibility chromatin information. But for this talk, I'm particularly focused on Signac and different kind of packages and functions that are related with that. One of the most relevant that we are going to explore is called peaks. We are also to explore feature metrics, region stats, and aggregate expression to pseudo-ball the chromatin accessibility object. But also one relevant function that is from the genomic ranges package that is called reduce. So you have here different resources that you can also explore and try to track what are the main, let's say, bullet points into this. And also I think that it was relevant to bring here some custom repositories that are very specific for particular analysis that helps in some way to solve some kind of technical issues. Because as I said, unfortunately, you don't find unique pipeline describing all these workflows. And also here, behind all these functions that I'm going to show, some of the functions, what really does is that they incorporate functions from other packages. They are wrappers of other particular, when they want to trigger particular functions. So one of these is MACS. MACS is wrapped in one of the Signac functions. It's not directly used to track peaks, But also I add here how you can install this environment in order to reproduce a particular analysis, and also the bed file format that is one of the main core things in this kind of analysis. Okay, so I will start with a general image of what we are expecting, of what we have when we are analyzing multi-ome data. So when we have 10x multi-data that has transcriptome profiles, we have RNA counts, but also we have in the ATAC side accessibility chromatin information. So we have information, or we have valid codes. We have like the green zone that is here in the middle for each one of these assays. So what CellRanger-ARC provides indeed is two different kind of counts. One arrow counts for RNA, and the other is peak counts for the chromatin accessibility information. So you don't have to do anything in special in order to have these two metrics. This is like the benefit to run the CellRanger-ARC pipeline, just like it is with default values. But let's say that in the real practice, we need to do a lot of things with the data if we want to improve the quality of them. So for this talk, we are going to focus specifically on the ATAC side. Now, just to provide a brief overview of this, let me make it a bit bigger. I have taken as an example this very common function in the Signac package that is called coverage plot. And what it does is basically to aggregate all the peaks in a way that we can visualize it in a way of genomic ranges, where we can see the peaks detected by the pipeline indeed. So this function, I want to use it just to depict what kind of elements we need to do this. So basically, all the pipeline works with several objects, but also we need genomic region that could be the name of specific genes that we want to track. Or we can also provide a range, that is a chromatin range, where we want to track specific peaks that are into that region. But also we can provide, for example, external peaks that were calculated with other pipelines, for example, with MACS, that is not part of CellRanger and is a wrapper that is included in Signac. So, for example, in this coverage plot that we have here, what we are looking at is in the first panel here, in the top panel, we are seeing in each one of the rows, there are tracks with a specific that has clusters or cell types specifics. And here for example, in each one of the tracks, we have peaks that were identified for the CellRanger-ARC pipeline. Let's say the metrics with counts peaks. And also here in the next, let's say, track, behind the cell-specific tracks, we have the gene that is this is a G ranges coordinates, when we can see let's say where it starts and when the genes ends. But also we can see here another like track that is for the peaks. So each one of the lines that we could see here, these small lines that we are looking at here, they are peaks that were detected by CellRanger-ARC indeed. So you can see here that some of the specific cell types have peaks, but others doesn't have these peaks. What is interesting here is that you can add an extra, let's say, peaks analysis that are coming from MACS. So we can have also another track here that is showing the peaks that were detected by MACS. So we can see here that, for example, the peaks from CellRanger and the peaks for MACS, in this case, match. We can see that we have here mostly the same peaks with the same white. But there are other cases when we have only peaks that were detected, for example, for MACS, and they were not detected by the CellRanger-ARC pipeline. So this is the play. You can, in an exploratory analysis, first take a look into the peaks detected automatically by the CellRanger-ARC pipeline, but also you can make your peak size, like taking into account different confounders, so you can have a different kind of peaks visualization, and you can validate if the peaks you are looking in the tracks are the real one or they have variability for some reason. So also when you have detected these peaks, The second thing that you want usually is to see if there is, for example, a relation between the gene expression of this particular genomic range and the enrichment on the peak. So here we have another track that is called links. And this link, what is showing is that we have a relation between this peak that we can see here in the genomic range and this other peak that we can see here in this other coordinate. So we can have distance relationships that are larger than others. This is usually related with specific classes of regulatory elements that we have available in the annotation. So in the ETF file, sorry. So here also other information that we have is that these links between, or these correlations between different genes and peaks also they are evaluated and they are attributed and scored so we can know what is the strength of this relation. So that way we can establish if we have co-accessibility regions at different distances, or what is the stronger relationship that we have. If these relationships are related with promoters, with enhancers, with silencers, depending on the analysis. So this is just like the big overview when we can track all this information here, but the challenge is how we produce this information, because you can produce this information since the beginning. The thing is that the interpretability at the biological level could be really different if we are following different steps. So this is what we are going to cover today. Okay, so the next point here is that, well, the big overview is that we have tracks, we have CellRanger peaks, we have MACS2 peaks, we have also linked peaks, we have the gene model or the coordinates of the model, and we have motif sites also. And all this information is related in a specific distance that is measured through the transcription start size, that is specifically the region when we are expecting to start the transcription. So what happened here? What happened here is that we have different ways to do this. For example, I'm going to focus on make evident that, for example, you can track peaks from CellRanger. We have two roads, the CellRanger-ARC peaks, or we can produce our own peaks. If we want to produce our peaks, we are going to need to use package like ColPeaks, that is the one that we are going to use as an example, that is wrapped in Signac. But the thing is that when you run ColPeaks, you have the power, for example, to add confounders. For example, let's say the major confounders in the detection of peaks is the cell types. So if you have very different cell types, you need to add the cell type as a confounder in your data. But also you can make different arrangements, like to add as confounder treatments versus conditions, or I don't know, whatever other, let's say, metadata that you want to track here. CellRanger picks what produce, it's a sort of data frame that is indeed a G-ranges data frame that contains information of the chromatin and on the start and endpoints of the peak detected. But you don't have a chromatin object. And if you don't have a chromatin object, you cannot compute the counts and you cannot run any kind of analysis. So to detect peaks with ColPeaks is just the first step if you want to follow this road. In the other, if you are taking the peaks produced for CellRanger and you have the separate object, you have counts with peaks and you don't need to do anything else in order to track, let's say, an exploratory analysis. Of course, you have a lot of advantages if you are trying to add confounders and you are trying to define distances and make a specific analysis. So in this case, you need to make a ColPeaks, let's say, to follow the ColPeaks road. So basically speaking, if you, in both cases, you need a chromatin assay, but you don't have one here. You only have coordinates with G-ranges. So we need to translate this information in a chromatin assay, and once that we have the chromatin assay, we need to run at least to compute three kind of specific analysis. One is to make corrections on the peaks. One of these corrections is called the GC content, and this is very relevant because this is related with the way that the transposasa works to identify these peaks. So it's well documented that the transposasa has preferences for the GC content. So these, let's say, these particular genomic regions could be enrichment, and so the detection of peaks could be like masked for this enrichment, or can be diluted in some way. So we need to do this in order to make a correction in the data related with this bias. Also, when you have this chromatin side, your data need to be normalized. And the most common way to normalize this data is through the TF-IDF approach for the ATAC data. And you need to standardize the data too. Well, once that you have your chromatin assay, it's QC, and you have like run the normalization, so you can run this kind of analysis. You can make differential accessibility between peaks, but also you can identify the correlations between different peaks, and you can do a lot of things. One relevant thing to mention at this point is that once that you have your chromatin object and you can run all the other functions related with this analysis, is that it's not mentioned in many of the tutorials that you need to evaluate the peaks. Because you are going to have peaks there, but not all the peaks are suitable or are not high confidential peaks. So we need to be very careful in order to understand that not all the peaks that are there are really, let's say, with the high quality that we need to run a specific analysis. So now, when we have, let's say, decide, okay, I'm going to run an analysis with following called peaks, but I want really to understand what is the difference between the peaks that CellRanger has and the peaks that the cold peaks generate. So these are really two different ways to analyze the data. So the best way to describe this is to say that when you are taking the CellRanger-ARC, the assays, let's say the peaks assay, run with default values, what you have is what we call global peaks. What that means? That means that you have peaks detected across all the cells, so we don't have peaks detected that could be cell-specific, cell-type specific. So it means that you likely don't have the same accessibility, chromatin accessibility information from one cell type than another. So this is really an average way to detect peaks. So you don't have this like information available for these specific cell types when you are taking the peaks coming from CellRanger. What happens when you have peaks this way? When you have peaks this way, we can run, for example, or we can try to correlate these peaks detected with the genes. So the basic approach is I load my peaks. I could also merge my peaks. I'm going to jump this step by now. I run the quality contours, I normalize, and then I trace my peaks. But look at here. You have only one genomic range for all the peaks that you have detected. You don't have a specific cell types for these peaks. If you look in the panel here below, you can see that I have 3 tracks. And these 3 tracks is because these peaks are specific for each one of my cell types. So I have peaks detected by this cell type 1, as well as the cell type 2 and the cell type 3. But this kind of analysis is not available if we use the global peaks. So that is why, under certain circumstances, we need to run the cold peaks and redo basically the peaks detection in order to have information related with cell type specific information. So these are here at this point, we have really relevant decisions to take, because if your data are expected to be very heterogeneous, likely you want to redo your peaks, or you want to also have an additional chromatin assay in your data in order to compare your global peaks across your local peaks. So to summarize, from here, I'm going to call the CellRanger-ARC peaks global peaks, and the cold peaks from MACS2 local peaks, because we have different information there. what happened next? What happened next here is
Sorry,
the
sorry to interrupt, Cynthia, but
yes.
can you explain to people that multiple samples are used at this step?
Here
Yeah.
in the Colpix. Okay. Well, here what happened is that let's say that you have in when you run the CellRanger-ARC pipeline, you run it for each one of the samples that you have. For example, if I have 5 samples, okay, let's say that I have 5 samples here. 1, 2, 3, 4, and 5. For each one of these samples, what CellRanger is doing, if I'm running individually, is that I have peak metric for each one of these. So indeed, I have a peak matrix 1, a peak matrix 2, and so on until the peak matrix 5. So when I integrate this data in order to identify, let's say, multi-ome clusters, what the merge function of CellRanger is doing is that it's indeed gathering all these peaks in one common matrix. So I'm not merging specific peaks detected for this one. So imagine that in the peaks 1, I have detected a peak here. But let's say that in the peak matrix 2, this peak is a bit, let's say, overlapped here. So this counts like different peaks in the global matrix. So we have one count here, and we have another count here, even when they are in the same coordinates. Also, what happened here is that these are labeled with a barcode that includes the sample when this sample is coming. For example, this is the sample 1, peak 1, and this is the sample, the barcode sample 2, peak, probably also is 1. So what you can imagine here is that we have a very large peak matrix containing overlaps peaks that are counted independently. So we cannot indeed to take the interpretability into this, we cannot infer cell type specific, let's say, interactions between peaks and genes, because the way that is integrated the data when we are doing or when we are using default values in the CellRanger-ARC pipeline. So in order to correct this is why we are using the CellRanger-ARC peaks, so we can have, we can recount the peaks, but say that the
You meant the MACS2, not CellRanger-ARC.
Sorry?
You meant MACS2, not CellRanger-ARC.
Yes, MACS2. Thank you, Leo. So what is doing CellRanger Colpix from is that it has a wrapper for MACS2 that when we can add and say that what we want is that the peaks counted needs to be specific for each one of the samples, but also for each one of the cell types specific that we have detected. So when I'm running the Colpix function here, what I'm saying to the command is that we want peaks that could separate the different clusters that I have in the data structure. I'm going to provide more details about it in the further slides. I have in total 10. So I hopefully it could be clear at the end. But this is basically what we have. Here we have a lot of data with independent counts of peaks, and we can here try to polish the peaks in order to try to have, we can do several things there, but basically what we want is just to add major confounders in order to have a clean peak metrics that we can use in further analysis. Why is the function to erase everything? Sorry. Okay, and okay, now just to take to this point, I know that there's a lot of information. I want to present here the basic approach when we are using MACS2 throughout the function Colpix from Signac. So the very basic approach is that we are going to load a separate object. And this separate object, we are only use the RNA counts of the transcriptome profiles, because we are going then to load the MACS2 peaks that we quantify separately, and we are going to create a chromatin object, correct data, and normalize it, and we are going to reattach this chromatin assay into CellRanger. why we need to do that is because if you want to run link peaks, we need information of the genes. So you cannot run link peaks only with the chromatin object because you need to correlate against the RNA profile. So you need both information. That is why you need to create the chromatin object and reattach it to the servant object. So once that you have this information, you can link peaks or make correlation between peak and genes, and you can also run differential accessibility between different cell types. So you can identify peaks that are only present in one cell type, but they are absent in other cell type. So this is the very basic way to run and explore the data. Now, when this is not enough, let's say, statistical power to identify suitable peaks for a specific analysis, we need to think in other ways to detect or to polish the peaks that we have. So talking about the Colpeaks results, there are two functions that I'm going to talk now. That is, we have the resource to merge the peaks in order to make more strongest peaks that can be deferred by coordinates. For example, let's say that you have here genomic range, right? You have a peak here, and you have here another peak. So when we run or when we want to build a shared peak set, what we are doing is to use this function that is here, that is called reduce. And what this function is going to do is just to merge these two peaks in just one peak that is going to merge these two genomic regions because they are overlapping. It's not the only way to do it because this function has, let's say, resources to also, for example, you have one peak here, but we have a small gap here. And this small gap, let's say, is insignificant for the kind of analysis we are running, or we are not interested in these small gaps. So you can say to the reduce that we can allow a small gap of, let's say, a specific number of pay verses. So we can have also here a big peak that is merging peaks with gaps. And all this happens in a specific distance. It's not arbitrary. You need to say what is the distance that you want from the transcription start site from the gene that you consider in order to merge peaks. So if we have a peak over here, let's say with the same gap, but it's not in the specific distance that we are setting, this peak is not going to be considered. It's going to be another peak. So you can play a lot here, and you can make this you can use these kind of resources if you want to compare, for example, data accessibility between different cell types. So let's say, as I said, these are different strategies. Any of these strategies is better than other, just they are different strategies. The other thing that you can do, you can jump this. This is an optional step. We can just go here, and once that we have our Colpeaks, I need to quantify the fragments. And for do that, I use this feature metrics function from Signac. And once that I have counted my peaks that are coming from Colpeaks, now I can create the chromatin object, and these chromatin objects, I can follow two roads again. If I try to trace link peaks, for example, and I don't have high confidence peaks, I have the resource to cell double these peaks. So basically, what I'm saying is that, for example, the most regular practice here that I keep seeing in different kind of resources is that in most of different investigations, they are only running cell double by cell type. So they only have these peaks detected by different cell types, and they can compare it. It's not really common to add here by sample, like is seen, for example, in the RNA profiles. Well, this is, let's say, the most common practice, but there is always resources to make it in a different way. Now, what I want to explain in these two layers, in the next two images, is why, or under what situations we can use reduce or we can use cell double. I think that is really relevant, and it's highly dependent on the kind of analysis that you want to run. So what happened here? Let's say that I have my peaks. I have drawn limb peaks. So basically, this image, what is depicting is that imagine that I plot my tracks. So I have a coverage plot here. In the coverage plot, I have here cell types specific. Let's say that I have 3 clusters, cluster 1, 2, and 3. And I have here one peak, I have here other peak, here other peak. I don't have peaks here. And here I have my link. I have here the gene model with the transcription star phi at some point here. And here I have my links. So I want to know what is the regulatory element that likely is driving the gene expression. So let's say that I have a peak that is running from here to here, where I have here overlapping of peaks. So this is, let's say, one of the information that I want to track. But even when you can see here peaks that are in some way linked, you need to measure how strong is the relation or the correlation between this gene and the peaks detected. So one way to do this is that we need to take the stats that were generated from MACS, and we need to add genomic regions. We need to identify the distance. And basically, we have 2 resources to do this. That is to run Spearman correlation or Pearson correlation. And you need to define what is the open window that you want to track, because remember always that we have a specific distance. So this is a very generous distance indeed. And usually let's say that the literature say that we have like 3 kind of scores to measure correlations. So let's say that the correlations that are under 0.1 usually are discarded peaks. So let's say that in order to create this kind of plots, when you want really to define the data that you want to interpret, you need to discard these peaks from here. So likely you are going to disappear some of these small peaks over here because they don't reach the minimum correlation score that you need. Links that have a correlation between 0.1 and 0.2 are called exploratory relations. So let's say that you can use it just to see what kind of possible regulatory elements you can find and this kind of hypothesis. And most of the papers, what they use is peaks that are greater than 0.2. So really, the peaks that you need to identify candidate regulatory elements are under this threshold. So this is very relevant because you can, at any point of the pipeline, you can make tracks. But the question is here, are your linked peaks relationships with a correlation score that is suitable for specific analysis? So you need to evaluate the peaks for know that. Okay, now what is next here? One that we have evaluated our peaks, and let's say that you don't find the suitable peaks that you want. So what kind of resources you have available? Well, you have 2 resources. One is I can merge the peaks. But how this helps to your analysis? Well, here I have another cartoon to explain this point. So the question is, should I merge my peaks or not? And sorry, this is not from Signac, it's from genomic ranges. This is an error. So let's say that, for example, these are your peaks. Before to merge your peaks, you have 2 different fragments overlapping in a specific genomic region. So when you do this and you want to, after that you merge your peaks, you have only 1 peak, as I explained before. And this is basically union peak sets that are in the same region. So basically it's collapsing and removing duplicate peaks. What happens if you don't do this? Well, you have different scenarios. I'm going to speak in the case when you have very different cell types. So what happens here is that if you are counting peaks coming from an external counter like MACS2, so you probably need to be careful with the double count inflation because for each one of these peaks you have a count. even when they are overlapping in a specific genomic region. So possibly you have a certain grade of differential expression bias when you are comparing the peaks. Also, if you have a lot of these peaks overlapping in the same region, you probably have a diluted signal because the redundant peaks can destabilize the normalization in the chromatin assay. Another possibility is that these larger but slower matrices, because these are very big, because you have one genomic coordinate but each one of these peaks, but it's going to take more size on this, higher memory and runtime to process the data. So another possibility is that when you are running limp peaks, you are going to have two peaks, but you cannot be completely sure about what of these peaks is, let's say, contributing more to the regulation of the gene. So many authors I have seen that they simplify these, unifying very overlapping regions in order to easy in some way the interpretability when they are analyzing correlations between peak and genes. Now, it's not also everything good, let's say. It's not an approach that we are going to run just because it's available. You need to consider some things. When you are merging peaks, all the metadata that were computed with MACS is going to disappear, because now you have different coordinates, you have different p-values, you have different fold chains. So the previous data that you have are not going to be available anymore, although you have specific arguments to keep the previous statistics and reattach to the object. But you need to do more things in order to keep this information. Usually, the authors that are using reduce, they are really trusting in the new genomic regions, and they forget about the MACS stats that they run previously. And this is also because it's for the kind of analysis that they are doing. Also, when you merge peaks, you can tell which groups contribute to the merge region. So you can have different overlapping peaks, but you are not really sure what of these peaks really contribute. There are also a function when we can measure overlaps, but let's say that this is a different kind of analysis. Also, you can expect that the genomic regions now are wider by definition. And well, if you are making this kind of motif enrichment analysis, you are not going to have available the submit information of the peaks. What that means is that, for example, you have here one peak. This is the submit information, possible related with information of motif enrichment. So if you are merging peaks, the two submits that you have previously disappear. So let's say that you have a weaker motif enrichment. So the analysis obviously is not focused in identify this kind of relations. Another consideration when making reduce is the differential accessibility between peaks could be challenging because you have merging regions. So you know that you can have specific gaps between peaks, so you can have overlappings that are not 100% matching. And finally, some reasons to skip reduce, let's say that you don't want to do it, is because your analysis is focusing in exploring cluster signals of peaks. So you don't need to have these, let's say, merging information about the peaks or to have the same system of coordinates, because you are only exploring peaks. Also, this is very common when you are integrating with other kind of data. Let's say different data sets coming from different projects. These are reasons to the use of this strategy that I previously explained. So as I said, you can opt to make these shared peaks, but you need to think on the different conditions that you have to do it. Now, the last point is, I should merge or not, I should make pseudoball or not in my data. So here, what I could say is that in order to decide if you want to pseudoball or not your peaks, it totally depends on your analysis goals, because what you are doing when you don't have a pseudoball is that you are going to focus your analysis at single cell levels. You are more interested in make this small difference that are between cell-to-cell variability. So you are not focused on regulatory elements, focus on different cell types, but mostly you are looking difference between cell-to-cell variability. So this is a different kind of analysis. Also, you do that because you want to plot some UMAPs to cluster these peaks in cell groups. You want to visualize accessibility for exploratory analysis, and you want to link peaks, correlations at peak level, so you have fine gradients of your peaks, but you cannot make direct comparisons between different cell types. Now, if you are thinking in a cell-level level, what you are doing here is basically you want to capture cluster-level differences. You want to try to trace regulatory elements that are present in different cell types. It's a lower resolution, but you can likely or carefully to identify this kind of relations. And where it's more robust if you have low counts or low depth cells. So it depends on data. It depends on a lot of things. So this is indeed, as you can see, a very conceptual approach. I'm just going to very fast to explain one script in 5 minutes, just to point specific steps. So we need CellRanger, we need Signac, we need the reference from the genome reference, we need the genomic ranges package to run reduce, and that's it. So the first step is that we are assuming that you have a CellRanger object with multi-ome clusters, let's say with nearest network clusters. So once that you run the CellRanger object, here in this step, we are going to load the peaks or the file with the peaks detected by MACS, right? That it was run with the call peaks function from Signac. So once that we load this file, this is a genomic ranges files containing, chromatin, transcription star size, and star and endpoints. Well, we need to extract the information that we need in the correct format that we need. So we can have the sequence, the ranges, the strand, and also we can have metadata related where the peak was detected. So I have here information. It was detected in thalamus cell types, for example. These are the stats that were computed by MACS. So remember, if you call reduce, you lose this, because you basically are merging different kind of coordinates. Of course, you can make something here to try to trace this information back. Also, in the case that you want to merge, let's say, peaks, so we use this function from genomic regions, reduce. I provide the G-ranges data frame, and I say here that I want to merge peaks that are between this gap. And with this command, I can retain some statistics that were computed by MACS as metadata. So later, in further analysis, likely I can use it for something. And next step, it's making some validations here that are not relevant. I load the CellRanger object because remember, I need the gene expression profiles to link to the peaks. So once that I run the CellRanger object, the next relevant step here is to define that I'm going to work with the ATAC chromatin information. So what that means is that I have the genomic regions, but I don't have the chromatin object, as I said. So we need to reveal a chromatin object. So we need to take the fragments from scratch from the CellRanger pipeline, and we are going to quantify with the feature metrics function using the peaks that we detect with MACS and the fragments that we have for each one of the samples. So these, at the end, are the count metrics that CellRanger-ARC produce, but this is the equivalent in MACS. So we need to do these steps in order to have counts. Once that we have counts, we are going to create a chromatin accessibility object again. So we provide the quantified metrics, we provide the peaks detected by MACS. Once that we have that, we need to reattach this chromatin object to the served object, but this is another chromatin object. I'm not overriding anything. I have available all these chromatin objects in the same served object. Now, once that I have this new chromatin object with peaks from MACS, I have to renormalize the chromatin assay. As I said, the usual steps to do this is to run the TF-IDF normalization. You can find the top features based in a specific threshold, and you need to run the SVD in order to standardize the data in the chromatin assay. Once that you have normalized the data, so now you can cell double if you want, providing the served object with the new chromatin assay. And I'm saying that my cell double is going to run in this particular size, in RNA, and the ATAC unified peaks that are coming from MACS. If you want to use another aggregation, you need to specify what chromatin assay you want to use. And lastly, you can make here your counts for cell double. Remember, this is another metric. So again, you need to create a chromatin object. You need to renormalize the RNA profiles from the cell double. You need to normalize again your chromatin object, but now for cell double, you reattach this cell double object to your data. And once you have all this information available, I'm going to skip some steps here. You can run peaks. You can make the correlations between the peaks that you have detected in cell double or in the MACS peaks or in the CellRanger-ARC peaks against your RNA transcriptome profiles with the genes that you particularly are interested. So you have here a lot of options to filter genes. And that's it. You have at least peaks. Here I omit the step to evaluate the peaks. But in theory, you need to make the evaluation to see if these relations, let's say, they have the statistical power to follow the analysis. And that's it. Thank you.